Hey everyone, welcome to chapter 12. So in this chapter, we're going to be talking about eukaryotic organisms that we study in microbiology. We typically focus on our prokaryotic organisms like bacteria and archaea, but there are a few eukaryotic microbes that we study in microbiology because either they are microscopic in structure or they are identified using microscopy. And so I'm bringing you into this chapter with a little story from your textbook about a disease known as potato blight, which occurred in 1845 to 1847. During this time in Ireland, potatoes were and not just Ireland, but all around the world, potatoes were the main crop. And they gave pretty much everything that you needed for nutrients that for people to survive. I mean, it wasn't the best nutrients. I mean, there are certain things that people would be lacking, but in general, they got a lot of their nutrients from potatoes. So every meal had potatoes associated with it. And then in 1845, blight hit. And so this is a water mold. This water mold, Phytophthora infestans, killed potatoes, decimated crops, and produced a famine that caused millions to die and millions to move. So there were about 1.5 million people who died at this point in history due to famine. So they starved to death. How scary is that, right? And then another over 1 million more left Ireland. So Ireland's population decreased by about 25% because of this blight mold. So this has happened periodically throughout history. So Phytophthora infestans is a little water mold that has caused blights at other times in history. This just happened to be one of the worst blights that we have seen. And so now I'm going to go into our eukaryotes because water molds are a eukaryotic structure. If I can get it to go to the next slide. Hello. Okay, maybe that. Nope, that did not work, did it? Sorry, hold on, there we go. Haha, I got it. All right, so let's look at our microscopic organisms, or our organisms that are eukaryotic that we study in microbiology. So there are microscopic structures called algae, but algae aren't always microscopic. They can be macroscopic as well. Algae is what you see in water in lakes when there's a lot of nutrients being dropped into the lake. Algae grow really well and turn the lake very, very colorful. Then we have fungi. Fungi are heterotrophs. These also can be microscopic or macroscopic. When I always think of fungi, I think of mushrooms. That's probably the most common fungi. Most people have seen mushrooms and possibly eaten mushrooms. Some people don't like them, I know, but I think they're delicious. Then we have protozoans. Protozoans are microscopic. They're always microscopic. They're single-celled heterotrophs. Protozoans don't fall into any specific category. They're hard to classify. They're kind of like if you've studied anatomy and physiology, we studied bones, and we have long bones short bones, flat bones, and then we have the bones that are called irregular. Irregular bones don't fit into the long, short, or flat categories. Protozoans don't fit into the plant, animal, or fungi category, so they're put into this group. We study worms, parasitic worms, that are macroscopic in general, but their larval form or their eggs are microscopic. And some of the worms actually can be microscopic as well. And then we study insects in this chapter briefly, just because insects are the organisms often that vector infection. And so we study them here. So when we talk about eukaryotes, remember that eukaryotic organisms have a nucleus with membrane-bound organelles. Eukaryotic organisms don't always have a cell wall, but if they do have a cell wall, they don't have peptidoglycan. They do have cytoskeleton, which contains some microtubules, microfilaments, and actin filaments. They can be haploid or diploid in number, whereas prokaryotic organisms are typically haploid. They can undergo asexual reproduction via a process known as mitosis, and sexual reproduction through a process called meiosis. And so in sexual reproduction, you have cells that are diploid, so they have a full amount of DNA, that reproduce or that replicate, and when they replicate through meiosis, they produce haploid structures that have half the DNA. These haploid gametes then can fuse together, producing a brand new organism that is unique from either parent. So we're going to go through these structures and talk about them a little bit. We'll talk about why we study them in microbiology, and we'll talk about their importance outside of just microbiology. So fungi can be found as molds, yeasts, or mushrooms. They are yeasts are always single-celled, and so they would be microscopic, whereas molds are filamentous, multicellular, and mushrooms are the structures that are reproductive for certain types of fungi. Mycology then is the study of fungus. So fungi contain a cell wall that contains chitin. When you hear the term chitin, you may think of a hard shell, because your insects and your ticks, they also contain chitin in their exoskeleton. That's the same structure that fungi use in their cell wall. Fungal membranes have a cholesterol-like molecule called ergosterol that helps to stiffen the plasma membrane, because remember, the plasma membrane is similar in all organisms. It's a very fluid molecule, so that ergosterol in fungi is going to act like cholesterol in animals, and it helps to stiffen and make that plasma membrane a little bit more stabilized. Fungi are heterotrophs, just like animals are, but they eat by absorbing nutrients, whereas animals eat by ingesting nutrients. So they excrete enzymes that degrade large molecules that they can then absorb. They are saprophytic, which means they take in nutrients from dead and decaying matter. So they're cleaning up our environment, which makes them very beneficial. They can be parasitic, which we'll talk about, And some types of fungi actually form mutualistic symbioses, which we'll also talk about. I'm going to stop here on this video since it's been 10 minutes, and then we'll get into the next video. Bye.